Hi everyone, I'm Nisarga N, a volunteer at Shivi Forensic. So, in this presentation, I'm going to cover the basics and steps of DNA isolation. So, without any further delay, let's get started. So, let us begin with the introduction first. As we all know, DNA is a deoxyribonucleic acid, which is the hereditary material present in almost all the living organisms. This DNA carries the genetic information that determines an individual's physical traits and biological function. So, what is DNA isolation? DNA isolation is the process of separating DNA from other cellular components such as proteins, lipids, and RNA. In the other words, it is the process of purification of DNA from the sample using a combination of both physical and chemical methods. The first isolation of DNA was done in 1869 by Friedrich Mischer. In general, they aim to separate the DNA present in the nucleus of the cell from other cellular components. So scientists use this DNA in number of applications such as introduction of DNA into cells and animals or also in plants or for the diagnostic purposes. On the other hand, mainly in the forensic science, it needs to cover the DNA for identification of individuals for paternity determination and plant or animal identification. Sources of DNA. Sources of DNA isolations are very diverse. Basically, it can be isolated from any living or dead organisms. So the commonly source that are used for DNA isolations include blood, saliva, buccal swab, hair, tissue, semen, which is a rich source of DNA in sexual assault cases, bone and teeth in the case of decomposed or old samples, and also in the urine, which contains the epithelial cells. In this, we have the limited DNA. The stored samples can come from archived tissue samples or frozen blood or tissues or exhumed bones or tissues. DNA isolation technique basically includes four major steps. First is the preparation of cell extract. Second one, the purification of DNA from the cell extracts, concentration of the DNA sample, and measurement of the purity of DNA concentration. So the first step of DNA isolation is preparation of a cell extract. So to extract the DNA from a tissue or a cell of interest, the cells have to be separated and the cell membranes have to be disrupted. For this, the collected biological sample is first washed to remove the external contaminants, and then the cells are treated with a lysis buffer containing detergents to break the cell membrane and the nuclear membrane. This process releases DNA along with the other cellular components into the solution, forming a cell extract. Chemicals such as EDTA, which is nothing but ethylene diamine tetraacetate, which removes the magnesium ions that are essential for preserving the overall structure of the cell membrane, and SDS, sodium diadecyl sulfate, which aids in disrupting the cell membrane by removing the lipids of the cell membrane, are included in the extraction buffer. Cell debris and partially digested organelles, etc., can be pelleted by centrifugation, leaving the cell extract as a responsible, clear supernatant. The extract contains DNA, proteins, lipids, and RNA, which are later separated during the purification. After the cell extraction, the second step is to purify the DNA. In addition to the DNA, the cell extract will also contain significant quantities of detergents, proteins, salts, and reagents used during the lysis step and RNA. A variety of procedures can be used to remove these contaminants, leaving the DNA in a pure form. The commonly used procedures are ethanol precipitation, phenol chloroform extraction, mini column purification. In this step, the proteins are removed by adding enzymes like proteinase K or by using organic solvents, and RNA is eliminated using RNase to obtain the pure DNA. Ethanol precipitation. Ethanol precipitation usually by ice cold ethanol or isopropanol. Since DNA is insoluble in these alcohols, it will aggregate together, giving a pellet upon centrifugation. Precipitation of DNA is improved by increasing of ionic strength, usually by adding the sodium acetate. Then phenol chloroform extraction. Phenol chloroform extraction in which phenol denatures proteins in the sample. After the centrifugation of the sample, these denatured proteins stay in the organic phase while the aqueous phase containing nucleic acid is mixed with the chloroform that removes phenol residues from the solution. Then lastly, the mini column purification. Mini column purification that relies on the fact that the nucleic acids may bind which is nothing but absorption to the solid phase depending on the pH and the salt concentration of the buffer. After purification, the DNA solution may be dilute. The DNA concentration is increased by precipitating DNA using cold ethanol or isopropanol. In the presence of salt and at the temperature of -20 degrees Celsius or less than that, absolute ethanol will efficiently precipitate polymeric nucleic acids. With the concentrated solution of DNA, one can use a glass rod to pull out the adhering DNA strands, while for dilute solution, precipitated DNA can be collected by centrifugation and redissolving in an appropriate volume of water. So this diagram represents the phenolchloroform method of DNA isolation. Initially, the sample contains intact cells. To break these cells, STS and proteinase K is added. STS acts as a detergent that disrupts the cell and nuclear membranes, while proteinase K digests proteins, especially the histones associated with the DNA. This mixture is then incubated to ensure the complete cell lysis and protein digestion, forming a cell lysate. After this, the phenolchloroform is added and the sample is vortexed and centrifuged. This step separates the components into the different layers. The upper aqueous layer contains the DNA and the middle layer interface contains the proteins and the lower inorganic layer contains the lipids and other debris. Finally, the aqueous layer is carefully transformed into another new tube as it contains the purified DNA which can be further processed for concentration and analysis. After DNA isolation, it is important to measure both the concentration and purity of the DNA sample. This is commonly done using a UV spectrophotometer or a nanodrop. DNA concentration can be accurately measured by UV absorbance spectrometry. The amount of UV radiation absorbed by a solution of DNA is directly proportional to the amount of DNA sample. Usually, the absorbance is measured at 260 nanometer. At this wavelength, an absorbance of 1.0 corresponds to the 50 μg of double-stranded DNA per ml. With a pure sample of DNA, the ratio of absorbance at 260 nanometer and 280 nanometer. Ratios of less than 1.8 indicate that the preparation is contaminated either with the protein or with the phenol. Thus, the measurement of DNA concentration and purity ensures that the extracted DNA is suitable for further forensic and molecular analysis. With this, I conclude my presentation. I hope you found this presentation informative and helpful. Thank you.